thank you to the organizers, and I'd also like to recognize the ophthalmology community. I am a cardiovascular local drug delivery veteran. Ophthalmology is new, but I've really appreciated the welcomeness and the collegiality. It's a fun group of people to be around, and I really appreciate the way you've supported us to date. At Interface, we made a discovery about 2 years ago, a way to engineer prodrugs with chemistry and with processing to create standalone, controlled release implants. These are our early studies, and you can see we started with coatings. Dexamethasone is what you're looking at here on the coatings that have these very beautiful controlled, essentially zero-order release kinetics in our in vivo experiments. We then quickly learned that not only were they coatings, but we could make them into solids like the cylinders, and that's where our intravitreal program began, as well as microspheres. In the microspheres, you're looking at triamcinolone acetonide as a prodrug engineered into a non-polymeric, no polymers present. Thank you. Thanks. And so when you look here, you're looking at a mechanism of action that is based on surface erosion. It lends itself to a very engineerable way of doing things. As the material degrades, the drug is released. So when the drug is gone, the material is gone. From an engineer's perspective, and we are a team of engineers, it gives us the ability to think about creating for lots of different indications based on what the true kinetics and desired product needs are. In terms of the other part to this is because there's no polymer, we can make these much more smaller than typically seen when you're taking a polymer with an API. So the form factors that you're looking at here, these are all dexamethasone-based prodrug materials, again, no polymer. The vision for the company is to be able to take this, and as we want to move forward with our dexamethasone intravitreal implant, continue to boil the ocean for other drugs beyond steroids for the purpose of treating as many appropriate indications as possible across therapeutic areas. Our strategy, we're starting on the steroids front across both ophthalmology and pain management and moving into other areas of ophthalmology as we go. This is our lead program based on our KOL feedback. We are targeting 6 to 9 months. You can see the implant in a rabbit eye there with 1/10 the delivery of dexamethasone that is in the commercial version out treating these disease states. Our needle is a 30-gauge needle, and again, no polymer carrier. This gives you a little idea of the data that we have to date. These are rabbit vitreous dexamethasone concentrations. The purple is the burst release of the PLGA dexamethasone commercial product. The orange is the data in the literature, just to highlight this is reproducible. And what you see in the red is the intravitreal IBE814 implant, steady eddy across 6 months. We still have cohorts of data that we are sacrificing to see the length of time. This is our VEGF study inducing vascular edema, and you can see that at 1 month, the IBE814 has a consistent profile, both fundus and fluorescein relative to the commercial product, and the difference between the negative control. As we get to 10 weeks, we hold with our ability to keep vascular edema quiet, whereas things change in the commercial version. When you get to 6 months, we're still holding with the ability to create benefit in the eye. This is how we see, this is, we've taken the PD and the PK, and you can see the check marks on the top highlight the extended sustained delivery from a benefit biology as the PK remains holding in the IC50 range. With the eventual, we'll see degradation of the material. We finished our preclinical early studies. We've got a positive read from the FDA, and we're now in the process of getting ready for the clinic in May of 2020. Thank you.
Thank you very much.